Hey, good morning and welcome everyone to this webinar. This session is going to introduce you to the next generation sequencing OmicsLogic training program. This program is going to be available in the context of OmicsLogic launch in Africa. Bioinformatics in Africa is an international initiative that connects multiple educational organizations that will be working together to bring cutting-edge bioinformatics resources to the community of students and researchers interested in learning and developing data science skill sets relevant to the current biomedical research, clinical R&D, and the biotechnology industry. So today we'll discuss the following topics. First of all, we'll talk a little bit about the OmicsLogic resources and different training platforms that will introduce you to bioinformatics. We'll also talk about breast cancer samples, an example where we can go from raw reads from the next generation sequencer and develop a table of gene and isoform expression. And that example will give you a snapshot of what this training will be all about. So as you understand the way this program is organized and conducted, we will then introduce the full curriculum and some details of live sessions that are going to be included in this upcoming program. Now, one of the major advantages of OmicsLogic programs is that we will introduce domain experts with rich experience and background in current biomedical data-driven research. So the participants of the program will have the opportunity to not only learn about bioinformatics and try to analyze data themselves, but also get answers about their research ideas and get mentor support to complete the courses, assignments, and effectively use the resources that we are going to be introducing. Towards the end of the program, we will also discuss how to register for this program and access these various resources. So before we go into the individual components of these topics, let me introduce everyone to our team and talk a little bit about how this program is going to be working. So the main organizer for this program is Dr. Mohit Mazumdar, who has a PhD in computational biology. And he's the director of business development and a mentor for the programs, helping to find the right match between the mentors and the participants of the program and make sure that all of the resources are being accessible and easily used by all of the participants. So our company, briefly, is based in the US, but we do have an international presence, working both in universities and with industry partners in Europe, the Middle East, and now in Africa. Our goal as a company is to provide cutting-edge bioinformatics research and training. And we work with several university partners to evaluate and refine the educational materials that we provide and make sure that they are effective and practical for the participants from different levels, including high school students, undergraduate students, graduate, and even faculty. Our mission as a company is to simplify bioinformatics and make it accessible, advancing research through the cutting edge and user-friendly next generation sequencing data analysis platform called the T-Bioinfo platform. We're working with over 5,000 students and researchers throughout our platforms to provide research support and big data analytic tools designed for biomedical data discovery. This all has been made possible due to the efforts of a rich interdisciplinary team, including the co-founders of the company, myself, Ilya Brodsky, and Dr. Alfred Tauber and Dr. Leonid Brodsky, who is the director of the Tauber Bioinformatics Research Center and an expert in bioinformatics and biostatistics. So what we will discuss today is also a result of hard work by some of the people mentioned here on the left. Julia Panov, who is an expert bioinformatician and helped prepare many of the materials we will use. Avi Titievsky, who is a computer scientist and leads the development of the T-Bioinfo platform. Joshua McKendall, who helps maintain the educational web portal that we will leverage. Dr. Harpreet Kaur, who is our OmicsLogic trainer and expert bioinformatician. And Bipsa Biswas, our community manager that I'm sure many of you have met already through the emails. So let's talk a little bit about OmicsLogic programs that we offer and how live sessions are going to help you leverage the materials that will be included in this program, including some online materials for asynchronous learning, like the hands-on courses, projects, assignments, quizzes, and the certificates. Number two, we'll talk a little bit about the analytical platform, the T-Bioinfo platform, which is a user-friendly implementation of complex bioinformatics pipelines and allows us to process, analyze, and visualize multi-omics data. And also, we'll talk a little bit about the data science component of bioinformatics, where you will learn about R and Python coding intended for biologists with minimal background to rapidly advance their skills in data science, to be able to understand data types, extract meaningful information by analyzing that data, and visualizing in preparation for publication and showcasing the results. So OmicsLogic programs are a result of close collaboration between Pine Biotech, which is a big data analytics company based in the US, and numerous academic institutions that have participated in content evaluation, curriculum review, and project design. As a result, the OmicsLogic programs have been thoroughly tested and are known to lead to excellent outcomes for students and faculty interested in bioinformatics, data science, and biomedical research. So you see some of our partners here. We've done a lot of programs with different universities, including the Louisiana Biomedical Research Network, Georgetown University, Amity University in India, and we have a lot of partners that are continuing to leverage our resources to provide bioinformatics experience and project-based learning to their students, as well as research partners that are leveraging some of these resources to conduct cutting-edge research. All of our online training leverages cloud-based technology to provide high-performance computing access. So we solve the problem of infrastructure, as well as detailed training resources that guide you to leverage some of these materials and tools effectively. It also makes the learning interactive, practical, and hands-on. So the two portals that we'll mention today are edu.t-bio.info and server.t-bio.info. These portals are interlinked, and you can already start by creating a free profile on this link, and the link is pasted in the chat. It's edu.t-bio.info. So if you can go ahead and go there, and you will see a way to log in. Let me show you how. Just go to the home page. So this portal includes resources that you can go through, starting from free resources, and anyone can already get started with Introduction to Bioinformatics, a course that provides you with an overview of what bioinformatics is and how bioinformatics is applied in basic research, biomedical projects, biotechnology, and agriculture. So I recommend anyone who has minimal background with bioinformatics to start with this course. Now, when you first get to this portal, you will see right here a way to log in. This is the link where you can also create a new profile, and you can do that starting with your free access to most of the courses that we're going to be discussing today. So right here, you can either log in, or you can register right here, or even connect with one of your social media profiles. Once you log in, you will have access to free and paid courses, and we have basic subscriptions that require minimal pricing. So they're priced at $10 per month, and you will have access to all of the materials that you will see here. You also get a personal profile, and I'll talk a little bit more about that in just a second. So that's the first portal, edu.tbio.info. It is your personal space where you learn bioinformatics and then start specializing in different subtopics, including metagenomics, transcriptomics, genomics, and others. As you go through the courses and you start applying what you are learning to data sets, you will leverage a user-friendly tBioInfo platform that allows you to use a point-and-click interface to study different data sets and extract meaningful patterns that could be interpreted from a biological perspective. So first of all, the basic membership on the edu.t-bioinfo portal provides you with 12 asynchronous courses that include the analysis of next-generation sequencing for genomics, transcriptomics, metagenomics, and epigenomics. So you will find those courses available, and you can either purchase an individual course or buy a subscription to have access to all of them. Inside each course, you will find a variety of topics that include both the biological side and the bioinformatics side. So you'll be able to learn the theoretical parts. And then you'll also be able to apply what you are learning to a practical project. Now inside, there's a navigation tool that allows you to get the most out of this, including covering different types of keywords that you'll be able to understand if you're not familiar with them, and also be able to study offline by downloading the PDF version of the lessons as well. Now, in this program, our goal is to not only tell you about next-generation sequencing and how data analysis could be used to understand the data, but also to be able to process and apply all of these to a project of your own. So in this program, we will provide both theoretical and practical training for those who are interested in next-generation sequencing that is becoming widely accessible and helps us study a variety of different biomedical and other types of biological topics. In each program, you will learn about the type of data, how to process it and prepare it for analysis, and utilize data science skills to mine and visualize large data sets and present your research to peers and experts for feedback. So a big part of what we do is make sure that this could be used by non-technicians and non-engineers. As you know, bioinformatics is a rapidly emerging and changing area. So you do have to have a lot of technical skills, typically, to process and analyze such data. In the absence of such experience, learning bioinformatics and applying bioinformatics tools to study actual data sets can become very complicated. So we've designed a platform that covers many areas of next-generation sequencing, mass spectroscopy, structural biology, as well as data science, machine learning, and data visualization that is intended not for only bioinformaticians, but also for biologists and clinicians. So we are hoping that if you are coming from a life science background or in medical school or potentially even working as a clinician, you're probably already recognizing the fact that next generation sequencing and genomic data has transformed our understanding of many diseases and many types of conditions. So using these tools, you'll be able to actually process and analyze data without any coding. How does it work? So you will find a user-friendly interface that provides different types of outputs that could be easily visualized and explored to understand what is going on in the dataset. Being complex datasets, you'll have to understand how these datasets have been prepared and what do they describe. But ultimately, without any coding, you'll be able to explore a variety of different types of datasets and understand what they mean. So remember that during this whole presentation, we do have a chat box. And so if you have any questions or you would like to have more information about any of the topics that I'm describing, please paste your questions post your questions in the chat box to make sure that I can address them during these pauses that we'll do throughout the presentation to take your questions and be able to respond. So if you have any questions about what we've covered so far, please let me know in the chat. And as you post your questions, I'm going to hand off this presentation to Dr. Harpreet Kaur, who is a trainer with OmicsLogic, and will explain how we will cover the material that I've described in the upcoming program.
Thank you, Ilya. So just let me share my slides. About me, my name is Harpreet Kaur and I have done my PhD in bioinformatics and I am OmicsLogic trainer at Pine Biotech. Today I will explain all the upcoming program and I will show you how we can use and analyze real NGS data using T-Bioinfo platform. As we are talking about real NGS data, let's look at a research publication from where we have taken this data. In the year 2016, there was a research article that is published in the OncoTarget, where Bradford et al. Have explored the whole transcriptomic profile of patient-derived xenograft models or PDX models as a tool to identify both tumor and stromal specific biomarkers. They investigated RNA-seq as a hypothesis-free tool to generate independent tumor and stromal biomarkers and explored tumor-stroma interactions by exploiting the human and mice compartment specificity of the PDX model. Across a pan-cancer cohort of 79 PDX models, they determined that the mouse stroma can be separated into distinct clusters, each corresponding to a specific stromal cell type based on omics profiles of the sample. In their study, they have explored different cancer types, including lung and the breast cancer subtypes. Complete publication is available at the following URL. In our program, we will try to perform various NGS analysis using the dataset from the patient-derived xenograft, that is PDX project. So let's understand what is PDX or the patient xenografts. PDX models are generated when a fresh tumor obtained directly from the patient and is implanted into the mice through subcutaneously or orthotopically into immune deficient mice. Authors maintain the principal histological, clinical, and molecular characteristics of original patient tumors while remaining biologically stable in the mice. Since PDX models are more closely resemble and recapitulate tumor growth in humans than standard in vitro cell line or cell line xenograft approaches, they remain key experimental platform for preclinical drug development. In our program, we will try to analyze two breast cancer subtypes, that is ER positive and triple negative. ER positive breast cancer subtypes is identified by the presence of the estrogen receptor on the cell membrane, while triple negative breast cancer is identified by the the absence of all the three common receptors on its membrane. This is just one of the differences between them. That is how it all changes. But it makes one is more treatable while the other is more aggressive form of the breast cancer subtype. In subsequent courses, we will using this T-Bioinfo platform that is already described by Ilya. And this T-Bioinfo platform to process big data sets, this platform is built for multi-omics integration and analysis of structured data with advanced methods like machine learning, mathematical modeling, etc. After you set up your account at T-Bioinfo platform, so make sure you try out this analytical platform. There are the different file format in which NGS data is stored. Let's understand one by one what is each file format means. FASTQ file, first FASTQ file provides us with the reads from the sequences. It is text-based file format that stores reads, nucleotide sequences, and quality score related to each nucleotide. Then the FASTA file is also a text formatted file. It represents DNA and protein sequences. Typically, this is the reference file for any genome or a protein. And then we come to the GTF file, that is genome transfer file format, which contains genome annotations. For example, GTF stores the start and the end of each genomic elements. It could be exon, isoform, or a gene. This analytical platform will provide you hands-on experience for anyone to apply learned skill to interesting data sets with color-coded logical annotation and explanation of the methods, then detailed input and output formats, reproducible workflows. So let me demonstrate how we can analyze raw RNA expression data and finally generate a gene expression table by using a pipeline on our T-bioinfo platform. So let me demonstrate that. So in our if we move to the T-bioinfo platform, there are the different analytic pipelines, and I am here using a demo pipeline that is where we have used a PDX project. Once you start this pipeline, you must have some files. Here, as we have already, this is a demo pipeline, so we have already uploaded the data sets, and you have to, these data sets provided in a form of the SVL files. So, and we have uploaded this data set and with the reference genome as well as the data set that you want to understand. Here we are using the PDX links for the PDX data set. On by clicking, I have uploaded this data set. Then the next step is Primomatic that removes adapters. This is pre-processing step. And once you uploaded your data, then you will perform this Primomatic step. Primomatic remove adapters. So just click on this Primomatic. You need not to know every coding, or you need not to do any coding program. You just have to understand your data and upload those data, and then you can follow these steps. Once you remove the adapters, the next step is you can remove the PCR duplicates using PCR clean step. So it is just taking a bit time. Okay. So PCR clean removes the any duplicates of these reads. Then the next step is the for the mapping. So you can perform mapping by different algorithms. So here we are using Bataille T2 that will ultimately to generate your transcript. So we to map these reads on a genome. So we will using Bataille T2. So about every single step, what it does, the complete explanation is given in each step. Whenever you going to perform any step, there is complete explanation on a platform. Once you have mapped the your reads with the genome, then the next step is to generate the gene expression table. So gene expression table, you can generate, it will generate your read counts as well. If you choose the normalized, that is PKM, then you can generate your normalized data in form of a table. So I am here only generating the cons. Then you can also perform differential gene expression and analysis, but I am not running here differential expression gene analysis. So I have generated a gene expression table here, and I am going to stop my pipeline here. So once to end the pipeline, once this pipeline is completed, you will find a various output files in a download pipeline folder. So you can check this folder, but are files that you have obtained. So you have obtained RSME expressions, PDX project preprocessing. So once I'm already downloaded this file, so I can check what kind of table I have obtained from the raw reads. So after running this pipeline, we are able to obtain a simple table where samples are present in the columns while the genes are present in the row. This is how T-bioinfo platform will help you to generate or to perform complex analysis using a simple manner. You do need not to have the knowledge of all coding and everything. So this is how T-bioinfo platform will help you. So this is the final table. So if you want to perform any kind of analysis, so you have already able to prepare this table. So now let me look at the questions if you have.
Okay, so I see there's a question. The question is asking, how does one know what is the next step in the pipeline? And could I briefly address this question? As you saw, Dr. Harpreet was showing a pipeline that every step of the way, a next series of options is highlighted, eliminating the most common problems or wrong selection that a typical new person in bioinformatics would make. So really, the platform guides the creation of the pipeline based on data input and the previous selection that you have made. But also, what we will discuss in the program and what you will find in the online training is that all of these different steps are explained, building your logic. And the color coding helps you establish that logic so you remember which color is associated with what type of logical step. For example, pre-processing, then mapping, then quantification is colored in blue, red, and green. So it helps you remember. And then next time when you try to apply it to your new data set, you actually remember that you have to take into consideration all of these different steps.
Thank you, Ilya. So let me go further. So in the
There's another question, Harpreet. The other question is, what kind of data do I upload for analysis?
This depending.
So, yeah. Well, let me briefly talk about different data types. Could I share my screen?
Yes, sure.
Okay, so there are different types of omics data available. And one of the things that we would do when we are doing this program is we will actually explore different types of publicly available data sets on repositories like the National Center for Biotechnology Information, NCBI. And we'll explore here how to find data sets related to cancer. So for example, if I go to breast cancer, you will find here thousands and thousands of samples that you can analyze, starting with, you can see here over 5,000 bioprojects just related to breast cancer. So we'll talk a little bit about how to find the right kind of data. And the benefit of the platform is that you actually don't have to download these types of data sets to your own computer. You can directly import them into a pipeline. So here you can see breast cancer. Let's talk as an example, tuberculosis, for example. You can find here transcriptional response to Mycobacterium tuberculosis in mouse peritoneal macrophages, right? So you can really find here information on a whole lot of different topics. You can see SARS-CoV-2 in patients with severe acute respiratory illness in Egypt, or SARS-CoV-2 from Brazilian Amazon. So we'll discuss how to find these types of data sets and how to then analyze them with the appropriate pipeline on the platform. Did that answer your question, Louis? Okay, excellent. Okay, should I pass it back on to Bipsa to explain how to register and find all the relevant materials on the program?
Ilya, Dr. Harpreet can take over. Then at the end, I will be going over the registration process.
All right, great. Thank you, Harpreet.
Thank you, Ilya. We believe that bioinformatics literacy is key across all stages of the data. Data generation, processing, utilization, and critical decision making. It is essential essential skills for lab technicians, researchers, and clinicians. A couple of months ago, we started engaging universities across Africa that showed a keen interest making such training available to their students and faculty, and resulting in this upcoming program. This upcoming NGS bioinformatics training program is a collaborative effort between Pine Biotech, USA, then the Department of Medical Laboratory Science and faculty of the Basic Medical Sciences Lead City University, Nigeria, also supported by Nigeria MedLab Congo, Lagos, Nigeria, a non-profit organization committed to the boosting the understanding of medical laboratory and biomedical data analysis. We will combine online sessions, self-guided study material, and practical assignments for an immersive experience that has proven to be effective in our well-known and respected OmicsLogic program. To support this program and guide you learning with us, you will have an opportunity to interact with a collaborative with an international team of experts specialized in the area of bioinformatics and data-driven research. In this one-month program, we will focus exclusively on the opportunities in the computational biology training and clinical research support. Our mission in Africa is to help a growing community of clinicians, biomedical researchers, university students, and industry professionals. So let's look at the schedule for the live session. These will be recorded as well mailed to the participants. We will start with an overview with the translational research and learn about NGS data and its applications, followed by the application of the genomics and the transcriptomics in the understanding the logics of the analysis. After learning about two data types by the online sessions and the courses that you will access through the T-Bioinfo platform, we will start applying statistics and machine learning in our example data sets. Once we have a proper, deeper understanding about application of some of these techniques, then we will look into the public repositories and that how we'll find our data and what data is available and where it is available and how you can upload these data sets on our T-Bioinfo platform to run any kind of analysis. So after learning about the data, the registered students will have access to start exploring the innovative research and educational platform, live sessions, and a team of expert PhDs in biomedical research, student mentor. Let's look at the details of each session. In this session that is going to learn, let's learn now more about it. This session will be dedicated to the translation research, where we will learn about the overview, tools, resources on the T-bioinfo platform, and the projects, the data sets, and the T-bioinfo analytical platform, introduction to the precision oncology, infectious disease, expectation, schedule review for the training program, and important deadlines. Then you have any Q&A sessions to address your questions. This program is designed to highlight the role of big omics data that is shaping modern biomedical innovation, where molecular data is used for precision diagnostics, big data integration, machine learning, and transforming personalization of treatment selection. And innovative algorithms allow for unprecedented capabilities with drug discovery, design of large molecules, and drug repurposing. The various types of omics data describes intracellular and intercellular processes associated with common disease. It can be studied to understand the role of various pathogens, microorganisms in our bodies and in our environment. And it also understands to pose various infectious disease and chronic risks that destabilize immune system and lead to cancer associated factors. The next session will be dedicated to the next generation sequencing, genomics, where we will going to study DNA variants and mutations. Under this session, we will be focused on to introduction to the next generation sequencing needs of the structures data, and associated bioinformatics methods. Then what are the different processing steps to generate germline and somatic mutation, and how we can perform various kind of analysis like mapping or to detect variants, and how we can interpret the data significance meaning from this genomics data, and how we can generate a significant mutation that are available in particular disease condition or a data condition. So to learn more in a detail, there are two different programs. Courses are available, that is Introduction to Genomics, Genomics 1. That's where you will find details regarding the genomics data and the deep understanding how these mutational data you can explore in terms of to study in terms of precision medicine, and how you can generate a significant hypothesis to find out what important and significant mutations are important. So these courses where you can learn detail regarding genomics. We will understand, we can analyze genomic data using T-bioinfo platform, and we will analyze the genomics data at mutational level using this T-bioinfo platform, where you will perform pre-processing steps. You will learn about how mutation to generate a mutation and how you can, what are different methods are available to pre-process these data. Then how can you align your reads on a genome to finally to perform a mapping or alignment, and how you can generate different variants calling, how we can generate differential variant calling using T-bioinfo platform, and how we can annotate these variant callings or mutations. Finally, we can analyze the post-processed data or the outputs using T-bioinfo platform. The next session will be focused on next generation sequencing, transcriptomics, gene expression data. So here we will learn how we can measure the functional proteins by the way of mRNA abundance and accurate identification of alternating splicing. an overview of RNA-seq, read quality and pre-processing, mapping on reference genome using various tactics and logics, and how we can align and alignment-free quantification, and how we can quantify the genes and the transcripts in a particular data sets. Then a summary of the structures data, and we will learn about how we can visualize the transcriptomics data using PCA and other approaches, and how we can finally elucidate a biological significance from this complex transcriptomics data. This pipeline, I have already shown its demonstration to you. So if you want to further go to for details, you try yourself once you have access to T-Bioinfo server platform. Next session will be focused on big data problem, that is using machine learning for the complex gene expression data, where you will learn about big data and complex collections representing individual variety requires new approaches for the analysis, and correlation between clinical manifestations of disease and molecule patterns that defines it. Then you will learn about machine learning approaches that allow computational power to apply to these challenges. And we will review the data exploration using dimensionality reduction like PCA. Then we will learn about clustering methods, that is unsupervised machine learning. And you will explore how classification and discriminative analysis of labeled data sets can help to detect a relationship in large scale data sets by supervised machine learning. So we will go through the unsupervised and supervised machine learning methods in this session. So then we have another platform that is for the coding and analysis. So where you can run your own scripts and you understand how these data sets are processed using different codes. So if you are interested to understand how these you can process, or you are interested to learn about your programming in terms of whether it's R or in the Python, so you will access to this coding platform. And we will also show you how different analysis approaches are done using coding platform. So let me look at some of the questions if you have.
Yeah, there are some questions. I think the questions are more related to the program training itself.
And I'll get to them in a while. Yeah.
Okay.
Yeah, so when we talk about the program registration and how to participate and what will be the timing and logistics, Pepsi can mention all of that. But I think that maybe you could show a little example with the coding. I think maybe that would be interesting.
We will come to complete that how you can register to this program. Just let me go ahead. So this is our coding platform, OmicsLogic coding, where different already lessons are available. So you can learn how visualization, how you can perform differential gene expression, how you can perform clustering, and how you can perform different machine learning methods. So these codes and the tutorials are available in coding in the R and the Python, and you can learn about that. In the next session, this session will be dedicated to the bioinformatics data resources and the databases. So like NCBI, SRA, GEO, RefSec, and other repositories of publicly available research databases, other like TCGA, NCBI virus, etc. So where the public data sets are available, this session is dedicated to that part. In this session, we will learn how we can extract biomedical data from the public repositories like GEO, SRA, etc. And we will also learn how we can upload these data sets to our T-Bioinfo platform for their analysis to derive any biological significance or interpretation. The last session will deals with the final exam and the bioinformatics research projects. Here you will learn about the planning of what your project proposal, how to present your scientific data, and how you can generate or make a hypothesis. Then there will be a question answer sessions are also available, and you can upload your presentation you give through the audio and the video, and then you can go through the case studies and publications and the data sets. So this program is project oriented with the real data, used cases and example adapted from high throughput, high impact publications. Projects are extracted from the relevant publications on the topics that fit the program and student profile. Publicly available datasets are curated to prepare focused assignments that will be illustrating how the data is processed and analyzed and achieve similar results to the publication. Alternative approaches that can perform a similar function will be discussed, and we provide a review of the methods section of the paper. Finally, the full data set is organized into a project format that can be reanalyzed and discussed by the student and the faculty. And you will also understand how you proceed for your own project once you able to perform analysis on already public data sets. Now I would like to pass it on to Vipsa, so that she can explain you how you can go through the registration processes and other parts. So Vipsa, please take a session.
Yeah, thank you, Dr. Harpreet. I will be sharing my screen now. Right. I hope everyone can see my screen right now. Let me know if you cannot see my screen.
Yeah, we can see your screen, but Vipsa, you have a little bit of a noise coming in. So if you can just make sure that your microphone's fine.
Yeah, okay. Is it better now?
Okay, yeah.
Yeah. Okay. So I wanted to talk about the different resources that you have access to as part of the program. So bioinformatics in Africa is an initiative by us, Pine Biotech, and our collaborators in Nigeria and Lead City University. So I will be giving you a quick overview of the bioinformatics for genomic medicine program that we recently concluded on September 20th. Right. So we asked all of our participants to give us feedback on what they thought about the program. And here is just a few of the participant feedback from Adidoin and Ekpen Yong. And this is what they thought about the program that we did, which was a short 1-week program. We also did a survey of the different participants, and we wanted to see who are the different people who are joining us. So we found out on this survey that we had 13% of students who came from undergraduate studies. We had 40% graduate students or who are pursuing the graduate studies. We had 29% postgraduate students, 15% PhDs, and 2% people who are from the industry. So this survey helps us to understand the kind of participants we have for a program, so that we are able to reach out to each of you, because everyone learns at a different stage. And this is how our resources, our mentor support is going to help you throughout the entire program. We also asked our participants about the omics technology they were more interested in. So 18% said that they were interested in learning about bioinformatics and multi-omics as a whole. 35%, which is a majority, wanted to learn more about genomics. 20% about transcriptomics, 8% metagenomics. Then you have epigenetics. 10% want to learn more about big data in biology. And there's 2% for immunoinformatics and proteomics. And there are others who have also want to learn about different things like agrobiotechnology. So we will be also talking more about that later. And this is the certificate which a participant receives once they complete a program. So we give out certificate of participation and certificate of excellence for each of our program. The certificate of excellence is based upon the asynchronous course results that a participant takes as part of the program. So I will take you through the registration process for the program in a minute. So this is the page that we have for Africa. It's called africa.omicslogic.com. I just posted it on the chat. So if you look at this page, you will be able to see that there are details about the program. You have all of the syllabuses given right here, the date of the program, each of the sessions, the topics of the sessions, and what are the broad categories that are going to be covered. And if you go down, you'll be able to see some testimonials, some of the courses which will be part of the program, and learn more about our team. So I know that a lot of you have questions about what is the pricing and how you can get started with it. So we have made the program available with 3 membership levels. So let me just talk a little bit about the membership levels. So you have a beginner membership level. So the beginner actually gets access to the full program. You get access to both the EduTBio platform, the server TBioInfo platform, the Code OmicsLogic platform as part of the program. And that starts at only $30 a month. And we also have intermediate. The intermediate means that when someone is doing a project, they can request for a one-on-one session with our mentors, while the advanced program, which is for $75, will also not only include mentor support, but also will include access to 1 more month for everyone to finish their projects. So this is a 1-month program, but for someone who is looking to work on their project, which may usually take longer than a month, they can continue to do so if they have this license. So this is the division. Now, to make everything easier for participants who are coming up, like students who are in Africa, we have a 20% discount code on each of these. So for example, if I select the $31, so I select the basic beginner license. Now I can enter my card details here, and if I go to the coupon, this is the code, take 33 off. I'll post it again in the chat and also email everyone. And if I click on apply, you will be able to see that you get it for only $20.10. So you can then continue to payments, and this is how you can register for the program. We also have another payment link for local payments in Niger. So if you want that link, you can request for that, and we'll be sending you that Paystack link. Or if you want someone to pay for you in Nigeria or in any other countries, we have our collaborators and you can request that. So for that, please email me at marketing@pine.bio for any kind of assistance regarding the registration process. And I will quickly move back to the presentation right now. So as part, okay, the early bird price expires by September 30th. So you have until like a week to register. The program would be starting on October 5th. So the early bird will be expiring by September. So just moving on, you can also come and join our community on both Facebook and LinkedIn. I'll be posting the links in a minute in the chat. And we have these groups where we have people from the industry, from the community, from different universities who have come and join us in both of these groups. So this one is in Facebook, and the OmicsLogic machine learning group is in LinkedIn. So please come join us. Thank you so much, Dr. Mohit. So if you have any questions right now, you can post them in the chats and we'll be taking questions. Can we enroll for the program after the program dates? We would not advise you to enroll for the program after the program starts. The reason is this, that you have to go the program, follow the program. As you see, there is a syllabus, right? So the syllabus has been designed to make sure that you are starting the program from a basic level and then you are going towards advanced. So you may join the program if you think you can make up. But if you say joining the program for the third session or fourth session, it will be difficult for you to follow through the program. So that is why I would not recommend it. But yes, if you want to, you can go forward.
But maybe it's good to mention that we also have other programs that are starting at later dates.
Yes. So this is also true. Thank you, Ilya, for that. So you can join any other program. So as you see that we have students who are, yes, Asha, I'll get to your question in a minute. So as I did show you that we have students and faculty, all of who has different interests. So we have programs running throughout the year. So we have different programs dedicated to RNA-seq analysis, metagenomic data analysis, data science. So you can join any of those programs later this year and next year. We have a list of programs that we can let you know that which are coming up. We also have the research fellowship that we offer for students. Currently, there are around 15 research fellows who are joining us from all around the world. So you can also be a part of that. If you don't want to be part of this program and you're interested in working dedicatedly on your research project, so you have that as well. Okay, so let's go back to the certificates, right? So, can you please mute whoever is? Okay, thank you. Right. And Asha, to answer your questions about the certificate, right? So the certificate that we offer are two different kinds of certificate. One is the certificate of excellence and the certificate of participation. So the certificate of excellence is given to anyone who is enrolled in a 1-month program who has over 10,000 points. So how do you get 10,000 points? You get 10,000 points when you do the different courses, when you do the different, run different pipelines and also do different lessons on the code OmicsLogic platform. So all of these would give you certain points that will add up to your final points, which can be 10,000. So people in a 1-month program can also get up to 20,000 points, given how much active they are. So this would depend on your own performance. And this is about the certificate of excellence. And certificate of participation or completion, rather, we would give it to anyone who has over 2,000 points. The asynchronous courses help us to understand whether a participant is actually involved in the program and how much they are learning, because the asynchronous courses supplement all that is taught in the program. If you would notice the schedule, then all of our programs are not, every day of the week. There are a limited number of gaps between each of the programs. So that is the time that you can rehash and refresh your learning of what you learned in the session with the asynchronous courses, run some pipelines on your own, come back to the mentors with any questions that you can have. So it's a well-rounded learning experience for you. Okay, thank you. If there are any questions, please let us know. We are already coming close to the end of the program. And Ilya, if you would want to add something.
Well, briefly about the certification, as we mentioned, the program material and content is reviewed extensively by our academic partners, but our major research partner is the Tabor Bioinformatics Research Center at the University of Haifa in Israel. It's a center dedicated to the testing and development of new algorithmic approaches for the analysis of different types of omics data. And they are also helping certify the program outcomes for our participants. So it's a joint certificate between Pine Biotech and the Tabor Bioinformatics Research Center. And we are right now working with a number of universities in Africa, or starting to, where we are hoping that we will develop joint certification in the future as well.
Thank you so much, Ilya. So if you all have any other questions, please post them in the chats right now. And we will be reaching out to you via email with the details of the program. And you can again always email us regarding registration or any other questions about resources. Okay, the 10,000 points again is based on the assessment and the activity that you do on the 3 portals. So all of our portals are interlinked with each other. The EduT Bioinfo portal, they are interlinked, and the Server T Bioinfo. So whenever you run a pipeline, you get some points. Whenever you do a lesson on code OmicsLogic, you get a point. Right. So I hope, Sani, that was helpful to you. If you have any other questions, you can let me know. For Neha, so if you're doing an asynchronous course, you would be getting the certificate right on the profile of right in your profile. So you have to look under courses, and then you will find certificates. So Introduction to Bioinformatics, Introduction to Genomics, and Introduction to Metagenomics all have their own certificates, which are automatically generated once you complete the courses. And you have to make sure that you click on each module and click on complete to make the course complete. If you just go through a course, the course won't be complete. So if you have any questions with that, you can again email me. Have any upcoming program regarding NGS application and bioinformatics in food safety? We definitely have many programs on NGS applications. One of our next program would be both metagenomics and transcriptomics, which are starting later this year. And regarding food safety, so we do not have something exactly on the topic of food safety, but all of our programs are related to NGS data. So you can learn from this program and apply the learning to your own analysis. So that could be an approach. If you have, again, any questions, you can email me marketing@pine.bio. Okay. So thank you everyone for joining today. We will see you all again, and please let us know if you face any challenges. Thank you. Thank you, everybody.